Hello, welcome to our single-cell multiome analysis series with SciDAP. In this video, I will demonstrate how you could start analyzing your data using CellRanger. The step called CellRanger Count will map your reads, generate QC metrics, and produce U-maps or t-SNE plots. You could name your sample here. I already gave it a short name. And you select the indices that we were generating in one of the previous steps. Then you need to attach your FASTQ files. If you have them on your computer, you could just upload it by clicking on the File Manager button and then upload. SciDAP also supports FTP connection, or alternatively, you could provide a URL if, for example, you are analyzing data from the geodatabase. In one of our previous videos, I showed how to split your data into proper FASTQ files when downloading it from the NCBI storage. If you have your samples uploaded this way, you need to provide the link. For that, you need to go back to your sample, click on the tab Files, and here are the files that are available in our FASTQ download output. So you could select the file that you need and copy the link address. Then you simply provide it here and click Attach. For RNA-Seq, we need 2 FASTQ files. And for ATAC-Seq, you need 3 of them. You do the same for every FASTQ file. Now ATAC. It's very easy if you know in advance what files you need. You could see I only copy some of the FASTQ files, not all of them. Third file, and the last one. Here we go. In the advanced settings, there is a button called Do Not Count Intronic Reads. In 10x multiome data, transcriptomic reads come from nuclei and most of them are unspliced. You could remove the intronic reads manually using this option, but it may reduce your UMI output as some reads could be potentially lost. Therefore, for single nuclear data, we recommend this option to be off. And this option defines system performance, and 4 cores is a standard. And you click your button Save and Run, and just wait until it's done. And in the next video, we will review the output of this pipeline.